We are pleased to present a video abstract of the FDA perspective and review article, Regulatory Implications of ctDNA in Immuno-Oncology for Solid Tumors. This is part of the special review series on liquid biopsies in the Journal for ImmunoTherapy of Cancer. In the era of precision oncology, ctDNA offers a minimally invasive approach to obtain valuable information about a patient's cancer without having to rely on tissue biopsies. There are a number of clinical uses of ctDNA in development, including for the diagnosis of cancer, to assess prognosis, and guide treatment decisions. Changes in ctDNA levels can be used to assess tumor burden and may provide lead time in identifying disease recurrence or assessing response to therapy over standard radiographic assessments. ctDNA is also emerging as an important biomarker and regulatory tool to support efficient clinical trials and drug development. For early-stage solid tumor cancers, ctDNA may be used to detect molecular residual disease, or MRD, to support early implementation of adjuvant or escalated therapy to prevent development of metastatic disease. ctDNA MRD may be used in clinical trials for patient selection and stratification based on the presence or absence of ctDNA after definitive surgery or radiation. Other trials may enrich for a high-risk population by only enrolling patients who are ctDNA positive after curative intent therapy. Adaptive enrichment may also be used to close a ctDNA negative arm at an interim analysis. In addition to this review article, an FDA draft guidance was issued in May of 2022, which further details our thinking on the use of ctDNA for early-stage solid tumors. Across small studies and pooled analyses, changes in ctDNA levels have been predictive of clinical benefit from therapies. Although not currently validated for use, ctDNA has potential as an early endpoint in clinical trials to support drug approvals. To establish clearance or decreases in ctDNA as an endpoint for regulatory use, additional data are required to correlate ctDNA with long-term outcomes such as disease-free survival and overall survival. Our review article discusses how meta-analytical approaches could be used to validate ctDNA as an early endpoint. Finally, an essential part of developing ctDNA for regulatory use is standardization and harmonization of assays and methodologies. It is important that all sites in a clinical trial follow standardized protocols for sample collection, storage, processing, and handling for consistent assessments of ctDNA. The timing of ctDNA collection in trials should be pre-specified, and potential differences in ctDNA shedding based on histology, grade, stage, and size of tumors should be considered. Assay validation is also necessary to establish the analytical performance of a test and encompasses the entire system from sample collection to output of the assay. Optimizing assay sensitivity and specificity involves establishing an appropriate assay cutoff. And in general, high sensitivity and negative predictive value are needed to support de-escalation of treatment, while high specificity and positive predictive value are needed to support escalation of treatment.